Welcome to the lecture series in Food Biotechnology. In this lecture, we are going to see how the biotechnology and its applications can be used to improve crops. We are talking about either increasing the nutritional level of the crops, increasing the amino acid content or the iron content or even a slight variation in the amino acid availability when you consume a particular crop, or you can change the nutrition component of the plant so that when we process that particular plant's food, there would be more bioavailability of the nutrients from that particular product. So this lecture is going to cover how various modifications have been done using the biotechnological tools so as to make it more consumer friendly. Changing the nutritional value of crops. One is the amino acid content of the proteins. An early application of biotechnology for improving the nutritional value of food has involved changing the amino acid composition of some common proteins of the human diet. In general, cereals have proteins with a low content of the essential amino acid lysine, threonine, and tryptophan, while the legume proteins have a deficiency of cysteine, methionine, and tryptophan. Among the most valuable sources of vegetable protein are the grain legumes. In one of these, example is soybean, genetic engineering has been applied to increase the content of the essential amino acid lysine in the seed proteins. Lysine synthesis in this species is finally regulated by a feedback mechanism. That is, when the lysine content is high, there is an inhibition of two of the enzymes involved in the metabolic pathway, leading to the synthesis of the amino acid. That is, the plant will synthesize lysine depending upon available presence of two other amino acids. It is a feedback regulation that as and when is required, the lysine amino acid content will either increase or decrease, and it is always maintained at a constant rate. The strategy concerned, or which has been used to improve the lysine content of soybean, is the integration of the genome of the soybean of genes from other species encoding the enzymes without the feedback mechanism. So normally in the normal cultivated soybean, you have a feedback mechanism. When lysine content increases beyond, the feedback mechanism will kick in, regulating the enzyme so that the lysine is not getting synthesized. So using biotechnological applications, what we have done is to remove the feedback inhibition so that lysine will always get expressed in the system. The transformation of soybean with the gene lysCM4. This was taken from Escherichia coli and the DAPA gene from the Corynebacterium encoding what we call as the DHDPS. Both are insensitive to feedback inhibition by lysine. So normally a threshold level of lysine inhibits the enzymes in the metabolic pathway. Hence, the forward mechanism, synthesis of lysine, stops. And when lysine content decreases below the threshold level, the feedback inhibition is lifted so that lysine synthesis happens. But when we use the genes which have been taken from Escherichia coli and Corynebacterium, these are not sensitive to the presence or the level of lysine presence. So, the transgenic soybean with the duplicated pathway of lysine biosynthesis has two pathways. One is the normal, controlled by feedback inhibition. The other one is insensitive, belongs to answering the transgenes, and it is insensitive to the presence of lysine. As a result, the lysine content of the transgenic soybean plants was over 100-fold the value of the untransformed plants. So the same strategy has been applied to other plant species like corn, wheat, and canola, and in all these, the lysine content has been increased. The second example where biotechnological tool has been used to modify the crops is modification of the fatty acid composition of triacylglycerol. As you are all aware, lipids are one of the main components of the human diet, and the consumer preference for plant-derived oils is increasing as detrimental to the animal fats. And among the plant seed oils, triacylglycerols are the ones which are very, very important. Properties such as the melting point, color, flavor, mouthfeel, spreadability, stability, and the effects on human health are very important when you regard consumption of fat. And for that, plant fats are much better than the animal fats. So all this is actually determined by the fatty acid composition of the triacyl glycerols. Common fatty acids in the commercial seed oils are the lauric acid, the myristic acid, palmitic acid, stearic acid, oleic acid, linoleic acid, and linolenic acid. Among these, oleic acid, this is a major monounsaturated fatty acid in the diet. This normally reduces cholesterol and LDL in the serum. So this is a good composition which we have to maintain. Transgenic plants overexpressing the desaturase gene that encodes for the enzyme catalyzing the conversion of the saturated precursor stearic acid C18 into oleic acid have been obtained. So first of all, the gene for the desaturase enzyme is identified, and this enzyme actually catalyzes the conversion of the saturated stearic acid to oleic acid. And transgenic plants have been created where the oleic acid content has been improved. The other pathways which have been important and which have been modified are vitamins and the other diet enrichment compounds. Vitamins are essential compounds for humans and other vertebrates, and normally these are obtained from the diet. And in addition, some vitamins are used as functional additives in food products. Ascorbic acid, or otherwise vitamin C, is used to prevent an oxidation in apples, peaches, apricots, potatoes, peanut butter, potato chips, beer, fatty acid, and oils. The carotenoids, that is the vitamin A precursors, are used as colorants in margarine, cheese, ice creams, pasta, juices, and beverages. Metabolic pathway modification to increase the vitamin A content. A typical example is the golden rice, which we dealt with in an earlier lecture. In rapeseed, transformation with the phytoene synthase gene has also increased in the level of vitamin A precursor, making it more vitamin rich. And in tomato, the transformation with the bacterial phytoene desaturase increased up to twofold the carotene content in the foods. Tomato already contains some amount of carotene, but that content was enhanced by the addition of the bacterial phytoene desaturase gene. Herbicide resistance. The first transgenic application to be widely adopted in agriculture was resistance to herbicides. Weeds are generally regarded to be the most serious problem to farmers, and it results in the reduced yield because they compete with the crop plants for water, light, and nutrients. Chemical herbicides are widely used by many farmers because they are cost effective and also are equally effective in killing weeds. Using single trait gene traits in transgenic plants, this particular methodology can provide a very specific way to protect the crop plant from the effects of a given herbicide. Herbicides generally work by targeting metabolic steps that are vital for the plant survival. For example, glyphosate kills the plants by inhibiting the production of certain amino acids that the plant requires for survival. This glyphosate is an active ingredient of the herbicide Roundup, which is marketed under the brand name Roundup. Thus, crops such as soybean and corn that have engineered to be resistant to glyphosate were given the name Roundup Ready. Let's see how this glyphosate works. This involves in the working of the shikimic acid pathway. We have the shikimate kinase, which actually converts shikimate to shikimate 3-phosphate. And then this shikimate 3-phosphate gets converted to 5-enol pyruvyl shikimate 3-phosphate, otherwise called as the EPSP. For this, you require enzymes. And then EPSP gets converted into chorismate. This chorismate finally gets converted into tryptophan and phenylalanine and tyrosine. So this step is catalyzed by the enzyme chorismate synthase. So when we see the working of the glyphosate works by binding to and inhibiting the enzyme 5-enol pyruvyl shikimate 3-phosphate synthase, EPSPS. This enzyme is active in the shikimic acid pathway leading to the synthesis of chorusmate-derived metabolites, including the amino acids tyrosine, phenylalanine, and tryptophan. The Roundup Ready soybeans were one of the first transgenic plants to be approved and produced on a commercial scale. Once they were produced at a commercial scale, they gained rapid acceptance by farmers. So what makes this glyphosate very acceptable to the farmers? One is this compound is readily taken up and transported throughout the plant, and the traits make it especially effective as an herbicide. Glyphosate is rapidly degraded by soil microorganisms, so it does not persist long in the environment after application. This is like biodegradable, so it is environmentally safe. It doesn't retain or persist in the environment or the soil, thereby harming the microorganisms which are present in the rhizosphere. So this is a benefit from both the environmental standpoint as well as in the crop management perspective, because farmers can plant any crop in a sprayed field relatively soon after the herbicide application. A second strategy to engineer herbicide resistance is to express a protein that will inactivate the herbicide if it is sprayed on the plants. So one, engineer an enzyme so that the pathway gets modified, or two, express a protein in the plant so that even if the herbicide is sprayed, it becomes inactivated because the protein will degrade it. This is the approach which is used in resistance against the herbicide glufosinate. This glufosinate is the active ingredient in the product marketed as Liberty. This generates a trait in crop plants which is called as the Liberty Link. This is the trademark of crops which have been engineered with glufosinate resistance. Glufosinate kills plants by inhibiting the plant enzyme glutamine synthetase, GS, which is responsible for the synthesis of the amino acid glutamine. As a part of the chemical reaction that produces glutamine, GS utilizes excess of the plant nitrogen in the form of ammonium that is incorporated into the amino acid. So when GS is inhibited in glufosinate treated plants, ammonium concentration inside the plant will rise to toxic levels. The glufosinate compound is naturally produced in some Streptomyces bacteria, in addition to having phytotoxic activity. Glufosinate also serves as an antibiotic because it is toxic to some other bacteria. Bacterial strains that are resistant to glufosinate produce an enzyme encoded by the BAR gene, which is called as phosphinotriazine acetyltransferase, or PAT. So this BAR gene was isolated from Streptomyces species, Streptomyces hygroscopicus, and this actually produces an enzyme which degrades glufosinate, and this has been transformed into several crop plants producing transgenic plants. The Liberty Link trait is currently used in transgenic corn, transgenic canola, and transgenic cotton varieties. Similar to the strategy in making Liberty Link crops, resistance to the herbicide bromoxynil, which is marketed or commercially available as Bactrile, was engineered by expressing the protein of the bacterial gene that will inactivate the herbicide. Bromoxynil kills the plants by inhibiting the function of photosystem 2, which is a crucial component in photosynthesis. So Brutracil resistant cotton is already very widely grown in US. Pathogen resistance. Pathogens such as viruses, bacteria, and fungi are severe and are a constant threat to agriculture. Multiple gene approaches have been used to create a plant disease control. The normal or the most effective way is to control the pathogens in a field is to use plants that are resistant to the problem pathogen. Resistance to a particular pathogen can often be conferred by a single gene, which is normally represented as the capital R gene. The product of this capital R gene, which is active, recognizes the presence of an active or a single virulence factor which is present in the pathogen. So product of the R gene will actually recognize the product of a pathogen gene. And in plant pathogen systems, this relationship is known to be gene-to-gene interaction. A typical example, bacterial blight is a destructive disease of domesticated rice, which is Oryza sativa, both in very widespread in Africa and Asia. and this is increased or caused by the environmental pathogen Xanthomonas zoraceae. Scientists looking for an alternative resistance, sources of resistance for this bacterial blight actually identified that gene in the wild plant in the Oryza longistaminata, which is native to Mali, which is resistant to the pathogen. But the detrimental factor is that this plant, the wild variety, has a very low quality and yield in terms of grain production. So the cultivated higher variety which has good yield and good quality, but it is sensitive to the disease. The wild variety, the quality is less in terms of grain production is also less, but it is resistant to the pathogen. So normally we will cross both. In normal breeding, we can have the hybrid, put the seeds for germination, then analyze all the products of the germination of that study all the offsprings from that study to find out which one actually carries the trait we are looking for. But using technological tools, we have identified an R gene called as the XA21 from the wild species. This gene has been introduced into wild domesticated rice using particle gun bombardment. And the presence of this actually very strong resistance to plants against different cultivars of Xanthomonas zoraceae. And although efforts of scientists scattered across the globe, the XA21 gene has been incorporated into several vegetables and several rice varieties of agricultural importance. So this is what we are trying to explain. You have the pathogen producing the virulence factor. So we are targeting, so this will target the virulence factors actually will induce certain level of defense in the plant. These virulence factors will target specific host proteins, and this R gene is produced which guards a specific host protein, thereby giving defense. So this is the strategy which has been applied. The protein products of R genes guard against the pathogens via surveillance of specific targeted host proteins. So when these R genes mediated defenses are triggered, the plant responds with a hypersensitive response and the rapid activation of defense gene expression. It has been known that normally in the case of viral diseases, if you are exposed to the virus once, then subsequently you will be resistant to again reinfection. This is also true as in the case with the plants also. So a previous inoculation with a virus can protect the plant from subsequent infection by a closely related virus. That is the principle of immunization. This form of immunization of plant has been known as cross protection, and it has been used so far for a wide range of viral diseases. Crop plants can be intentionally inoculated with a viral strain which is very mild, with the hope that this will protect the plant against future viral outbreaks. In the cases of inoculating with wild or mild strains of plant virus, there is always a chance that inside the host, these mild strains can become more virulent. Or it can also produce a drag, thereby reducing the level of protection or defense molecules which are already existing. With this advent of the genetic engineering of techniques in plants, it has become possible to express just a portion of the plant viruses within the host. This approach led to the resistance in some very closely related species of plants. Most plant viruses are relatively simple in terms of either the genetic makeup, consisting of very few genes, either coded by RNA or DNA, which the genome can be either DNA genome or an RNA genome. By expressing a portion of the viral genome constitutively in plants, a system of specific targeting of incoming similar RNA sequences which can be activated in a potential host plant. The RNA silencing mechanism is very active in certain plants, including humans, and it has also survived and evolved partially as a surveillance protection against viruses destroying or viruses becoming more virulent. A great successful story with this regard is the RNA-mediated viral resistance, which has developed in the production of papaya in Hawaii. The entire population, almost the entire population of papaya, cultivated papaya in Hawaii, was threatened in mid-1990s by the spread of a virus which is called as the papaya mosaic virus. By expressing the code protein gene of the mild strain of the virus in papaya, transgenic plants were created which were resistant to the incoming pathogenic viruses. And these varieties of transgenic papaya was first introduced commercially in Hawaii in 1998. And so far, all the transgenic lines have remained in good condition, and they also express the viral resistance character. This process of silencing or getting resistance to the virus was by a strategy which is called as RNA-mediated gene silencing. So first step is to make the virus-resistant plants, and a portion of the code protein gene of the PRSP was transformed into the transgenic plant, and it was also expressed in the papaya fruits. Following transcription, the RNA targeted and hit targeted sequences and sequence-specific degradation of the similar RNA sequences that is found on the incoming PRSV viral genome. The initial degradation of RNA is carried out by an enzyme which is called as dicer, and the process is mediated by an enzymatic structure of RNA and induced silencing complex which is called as the RISC complex. Ultimately, this will lead in silencing, that is, the RNA will get cut or cause block of transcription, or it will also can involve or fail the translation process also. Modification of plant oils. The fatty acids which are produced by plants are the sources of oils used in food, used in cosmetics, in detergents, etc. Oilseed rape or Brassica napus has been used as a source of oil for many years. Canola is the scientific common name for this, and it has also been known that canola oil has low levels of the harmful glucosinolates. So we now have transgenic plants or transgenic canola with a thioesterase gene, and this was taken at the California bay tree. The oils that accumulate certain higher levels of beneficial fatty acids. The bay leaf thioesterase enzyme expressed in canola causes premature chain termination of growing fatty acids. So when the fatty acid chain elongates, because of the presence of this thioesterase enzyme, the fatty acid elongation stops in between. So this will result in the formation or the accumulation of a 12-carbon compound, that is the lauric acid, and the 14-carbon myristic acid instead of a long chain containing 12 plus 14 or that many number of fatty acids. These short-chain fatty acids, which make up the canola oil, make it much more suitable as a replacement for palm oil and coconut oils, and especially in products such as margarine, shortenings, and confectionaries. The second example is soybean oil. By decreasing the levels of the enzyme called as D12 desaturase in transgenic soybeans, the oleic acid content of soybean has been reduced. And to decrease the levels of the enzyme, enzyme expression, the normal soybean gene, which is FAD2 gene, which encodes the D12 desaturase, is repressed using what we call as gene silencing. And a second copy is actually introduced into the plant. So the first gene is silenced and the second gene gets overexpressed. That is, the endogenous plant gene is silenced, whereas only the bacterial gene or the transgene is getting expressed in the transgenic plant. High oleic acid soybeans, which are transgenic, they have low levels of saturated fats and trans fats. So this means that the need for hydrogenation process, which is used to make soybean oil suitable for food like margarine, will be replaced. So you are going to land up with a healthier product. This also keeps the oil in a liquid form and make it more heat stable for cooking applications. Hence, all these different, so far we have seen 3-4 examples where the tools of biotechnology and its applications have been found to be very vast These are only a very few examples, but like this, we have potentially a huge number of applications. So as a questions for this particular lecture, what I'm going to ask is, what other potential applications of biotechnology and its tools can be used in the improvement crops? What are the characteristics you are looking for? And when you make a food product out of that modified crop, what sort of processing would you use? What sort of conditions would you have to standardize? Thank you.